Plants lose water by evaporation through a process known as transpiration. In transpiration, the stomata on leaves open up so that water may be released. The stomata are triggered to open in light environments. It is predicted that light environments will cause a higher rate of transpiration in a plant. Through the process of a stomata peel and determining the number of stomata on the leaves, plants are tested on their rate of transpiration in two separate environments, light and dark. After 5 days, the results showed that the plant in the light environment lost more weight, proving a higher rate of evaporation. The data concludes that plants in a light environment have a greater rate of transpiration. Transpiration is the process through which water is lost from a plant by evaporation. Water is taken into the plant through roots and root hairs by osmosis, and it exits the plant through tiny openings on the underside of leaves known as stomata. Oxygen and carbon dioxide are exchanged through the stomata. Stomata are triggered to open in light, so carbon dioxide is available for the light-dependent process of photosynthesis. Stomata are closed in the dark in most plants, although even the slightest bit of light can cause stomata to open, such as the light of dawn. The number of open stomata as an effect of light-dependent reactions have an increasing effect on the rate of transpiration. Therefore, it is hypothesized that if a plant is in the light, then there will be a higher rate of transpiration per square centimeter, because the light-dependent process, photosynthesis, requires a stomata to open up, which will further lead to a higher rate of transpiration. Based off of the AP Lab manual, the number of stomata on the entire plant was calculated. The whole plant method was used to find the rate of transpiration. The two plants were watered and placed into a plastic bag in order to avoid evaporation from the soil that would change the results of the experiment with weighing the plants. The environmental factor that was chosen to affect the rate of transpiration was light versus dark. One of the plants was put under light while the other was put in a dark drawer. They were weighed the first day and each day following. Beginning with two plants of a similar weight, the plants were placed in separate conditions to be observed by the different changes their individual weights would undergo because of their transpiration success or failure throughout a 5-day period. During the experiment, it became clear which plant was able to successfully transpire compared to the other. When placing two separate plants of the same kind in two different conditions, one being in complete darkness while the other under a light, the group observed which plant would transpire more. After the experiment was completed, the group reviewed the results and looked back on the original hypothesis to determine the accuracy of its prediction. It was predicted that the plant under the light would transpire more because being under a light, the plant's stomata would be open and able to complete the process, as opposed to that in darkness where the stomata would be closed and unable to release the water vapor created during the process. In conclusion, transpiration was greater in plant 1, which was under the light, while the plant 2 in the drawer still transpired but at a much slower rate. The leaves on plant 2 were wilted, most likely because the water was in the soil trying to sustain the root system more than the shoot system, because the root system controls the storage of the nutrients in water. It was learned that in order for transpiration to occur, the stomata have to be open. The number of stomata found in the beginning of the lab critically determined the rate of transpiration that was concluded.